Thank you and good afternoon, everyone. I appreciate you taking the time at the end of the morning here to come and give me the opportunity to give you an update on Caladrius Biosciences. We are a publicly listed company on the NASDAQ in the United States, developing late-stage products in the area of cardiovascular cellular therapy. As with most public companies, I will be making some forward-looking statements during the course of my presentation. So if you're making investment decisions based upon what you hear today, please do keep that in mind. So what is the investment thesis behind Caladrius? Well, we are a platform company based on the use of naturally derived CD34 positive cells for application in the treatment of ischemic diseases. That platform has been used to derive 3 advanced stage clinical programs, 2 of which have received the equivalent of a breakthrough designation in their respective areas of development jurisdiction. Our products benefit from a very substantial intellectual property portfolio, and we are focused on indications which really represent very large unmet medical needs around the globe. As an investment opportunity, we offer over the course of the next 12 to 18 months a number of value-creating events across the entirety of our pipeline. We are led by an experienced management team, which I will introduce in just a moment. And we have, for a company of our size, actually a relatively stable financial situation, which I will also detail later on. So as you can see here, just a brief summary of our executive management team, a group of people that collectively have more than 100 years of international drug development experience and with substantial domain-specific experience. I will point out that our Chief Medical Officer, Doug Lasordo, is actually one of the founding fathers of CD34 cellular therapy when he was an academic at Tufts University, and we're fortunate to have one of the world's leaders in this technology as part of our management team. We also benefit from the advice of what I affectionately call the cardiovascular mafia. This is the group of cardiologists internationally which really need to be supportive and involved in your programs if you expect to get any kind of traction at all. So let's talk about the technology platform briefly. I think that those people who have any experience with cellular therapy may have had some bad experiences. They have a knee-jerk reaction where they simply say cellular therapies don't have mechanisms of action. It's all magic. Nobody knows what's going on, and therefore we want to shy away. Then they talk about the lack of clinical data, since so much in cellular therapy has been announced based upon very small cohorts of patients. And then there's the question of the reproducibility and the economics of manufacturing. And then finally, whether or not you can actually have any kind of intellectual property, any exclusivity in this area. So I will address each of these very succinctly going forward as it relates to our CD34 cellular therapy platform. First and foremost, CD34 cells have a very well characterized and documented mechanism of action. They are naturally occurring endothelial progenitors. They function to promote angiogenesis of the microvasculature, and that's exactly how we use them. And this has been demonstrated multiple times in animals and in humans. As it relates to our clinical database, we're now approaching over 1,000 patients exposed across a multiplicity of indications done by many investigators at many institutions in many countries. So this is not a technology that only works in the hands of a single individual. And what's interesting about this clinical database is that in every case, there's a consistency of findings. We show that a single treatment elicits a durable therapeutic effect. That's why we can refer to these treatments as curative. And we have never reported a cell-related adverse event in any study. As you might imagine, from an autologous cell therapy, people receive their own cells, so there's very little chance of immunogenicity. On the manufacturing front, we have a very simple process. It is 4 days from vein to vein, as we say. The donor is in fact the patient. The donor receives several days of GCSF therapy to promote the migration of their CD34 cells from the bone marrow, where they naturally reside, into the peripheral blood. A sample of those monocytes is then harvested through apheresis. It's sent to a central processing facility where the cells are sorted, concentrated, and then resuspended in a proprietary media, and then sent back to the facility where the patient receives the cells as part of their treatment. The site of injection and the volume of injection are dependent upon the indication being treated. There is no ex vivo expansion. There is no genetic modification. So this is a very simple and straightforward process. And it's already now been scaled up to the commercial level and transferred from a technology perspective to 5 different manufacturing sites around the world. And then finally, from an intellectual property perspective, you can in fact patent cellular therapies. As the slide demonstrates, we have patent protection out to 2031, and the combination of claims in the granted patents permits us to have something what we would call the equivalent of composition of matter for a cellular therapy, making it virtually impossible for someone else to harvest CD34 cells and use them in a manner similar that would not infringe upon our intellectual property portfolio. So now let me take a few minutes and just speak briefly about each of the lead programs. We have a program in critical limb ischemia, CLI, which is in registration studies in Japan, a program in coronary microvascular dysfunction, CMD, which is beginning phase 2 in the United States, and a program in no option refractory disabling angina, or NORDA, which is phase 3 ready in the United States. The first of the programs is the program in Japan, and you may ask, why is a US company beginning development of their technologies in the Japanese environment? It's because at the time that we started clinical development, the Japanese regulatory authorities were the most advanced in providing an accelerated path to registration for regenerative medicines. We have in fact been awarded a Sakigake designation in Japan. It's the equivalent of a PRIME designation here in Europe or an RMAT designation in the United States, and it effectively awards you the benefits of both a breakthrough and a fast track designation from a regulatory perspective. This program also has an ATMP designation here in Europe. For those who don't know, critical limb ischemia is a disease of obstruction of the arteries in the lower limbs. It manifests itself as severe rest pain, debilitating rest pain that requires narcotics for treatment. It is also manifested by unhealing foot ulcers, ulcers that can remain open and festering literally for years, and often then degenerate to a gangrenous condition. The prognosis for CLI patients is poor. They often go to multiple amputations and often to death as a result of the disease. And for those who may doubt the severity of the disease, I just offer this slide taken from the literature, which shows that except for lung cancer, CLI is more prevalent and more fatal than every other cancer that is known. So this is a pretty important disease and requires significant advancements in treatment, because no option CLI offers no pharmacotherapy treatments today. Now, we are treating those patients who are considered moderate to severe, Rutherford 4 and 5 on the severity scale. And this shows you an example of how we can transform those patients from CLI to CLI-free with a single treatment. And over the course of about 6 months, you see about 85% of the patients converted, and they remain converted CLI-free for the duration of the trial follow-up, which was 4 years in this particular case. Again, a truly regenerative medicine. The laser Doppler image also gives you a sense of how the cells reperfuse a foot which was starved of blood, and therefore with tissues that were dying. A similar trial in the United States using the same dose and the same technology in the same population shows that we can meet the other common regulatory endpoint, which is amputation-free survival. And you can see here in a relatively small study, we were still able to achieve highly statistically significant amputation-free survival. And that has led to this trial here, which is being conducted in Japan. It was designed in conjunction with the Japanese regulatory authorities, and it is registration eligible. It is a trial for which we expect to have enrollment completed in the first half of this year. Top line data by the end of this year or early next year, a JNDA filed by the middle of next year, and with a 6-month review clock, an approval in Japan as early as the end of 2021. And since we're doing everything with the same cells, with the same population, the same dose, the same method of administration as was done in phase 2, we believe this has a very high probability of clinical and regulatory success. And we're in conversations now with potential partners to commercialize this product in Japan for us. Because it's an open label study, I can show you the results from one of the cohorts, the Berger's disease cohort, which is a type of CLI, an orphan condition. And you can see here, we had originally 5, which was then changed to 6, and now 7 because of compassionate use. But Berger's disease patients almost exclusively deteriorate to multiple amputations and then to death. And you can see here that we have converted 50% of the patients in the trial to CLI-free, which is a highly significant outcome and will likely lead to a label in Berger's disease for this product. Our next program is in coronary microvascular dysfunction, and this section of the talk deals with the cardiovascular applications. And I take a moment just to remind everyone that if you die of cardiovascular disease, you're just as dead as if you die of cancer. Most people seem to forget that. And in fact, cardiovascular disease remains the #1 cause of death in the developing world, and multiple studies, including one that was just released at AHA in November of last year, indicate that interventional techniques, available pharmacotherapies, and bypass surgeries do not, I emphasize, do not improve survival in patients with cardiovascular disease, nor do they prevent second heart attacks. And so there is room here for treatment that applies in a different direction, and we believe it's the microvasculature that needs to be treated. In the disease of coronary microvascular dysfunction, patients fail a stress test, have severe and debilitating angina, but upon angioplasty, they find that there are no large vessel lesions, no large vessel disease to treat. It's a disease that we believe, based upon statistics, affects millions of people worldwide, probably about half of the people with any type of coronary arterial disease, and it tends to afflict females more than males. No one seems to know why. We ran a study that we just reported at AHA in November that was using our cells to treat these patients, measuring coronary flow reserve as the endpoint along with the symptomatic scores. It was done at Cedars-Sinai and the Mayo Clinic in the United States, and we were able to report remarkable data. For the first time, a treatment was shown to convert patients with coronary microvascular dysfunction to CMD-free status. That means we've changed the physiology of the cardiac muscle, and we had a corresponding highly statistically significant improvement in symptom scores across the board for these patients. So this is a program now that has generated quite a lot of excitement. It'll be moving into a randomized phase 2 program later this year, and we will be raising some money in order to support this program into phase 2 and looking forward accelerated development in the United States and Europe. And then our last program is in no option refractory disabling angina, a disease similar to CMD, except these patients also had large vessel disease, which has been treated. This is a program for which we've generated quite a lot of clinical data, and this is in fact the phase 3 ready program in the United States with an RMAT designation. I'll just show you some of the randomized phase 2 data using the clinical endpoint that the FDA requires for approval. You can see here with 168 patients, we were able to easily demonstrate statistically significant improvement in exercise time at 6 months, which is the endpoint, which persisted to 12 months. A reduction in angina frequency at 6 and 12 months, and perhaps most importantly, a mortality benefit at 2 years, which is continuing to diverge. So we are in fact able to prevent death and prevent second heart attacks in the case where interventional techniques was not able to do so. We have negotiated a phase 3 protocol, a single protocol necessary for registration in this indication with the FDA. It's 400 patients, and it's a protocol that we will start probably sometime during the course of this year, either once we've achieved a partnership for the program or upon some significant additional funding. It's a $70 million program and will take 39 months from first patient in to top line and data in an indication where there are absolutely no available therapies for these patients and where there is a very large market opportunity. So as I mentioned, across the pipeline, a number of milestones that could be value generating throughout this year and into 2021. We are a company that has actually a stable financial situation. We finished the year last year with about $25 million in cash. We expect an influx of non-diluted cash during the first quarter based upon a large grant that we've been awarded by the state of New Jersey. And we have zero debt. Very simple capital structure, and we'll be looking to increase our shareholder base to advance the phase 2 and phase 3 programs as I described. So this brings us back to where we started, a platform company, a number of advanced stage clinical programs, an experienced management team, which over the last several years has demonstrated the ability to advance these products, and a stable financial structure. And we look forward to seeing you join us in this quest to bring new treatments to patients who have no recourse at this point in time. I'm happy to take a few questions. Thank you for your attention.